This is lecture 21 for Biology 177, covering membrane potentials. Now, we finished the last lecture, lecture 20, talking a little bit about how we produce electricity in our body. To review, electricity is accomplished and produced through membrane potentials. This means that we have a movement of different electrolytes or ions, which are charged particles that move across the membrane. Remember that cations are positive and are sodium, calcium, and potassium. Anions are negative ions, and that includes mostly chloride, but also the proteins that are going to be inside your cell. Now, cations and anions are balanced in the body. So throughout the entire body, your net charge is zero. However, across the cell membrane, it's going to be unbalanced. And this is due to the proteins that are inside the cell. So now that we know what cations and anions are, we can talk a little bit more about membrane potentials and the actual environment that we have inside of our body. Now, membrane potentials are resulting from the unequal charge distribution across the cell membrane. So the inside of the membrane is slightly negative, and the outside of the membrane is slightly positive. This is the characteristic of all living cells. Now, this results from a few different things. First, the difference in composition of the cytosol, or the intracellular fluid, and the composition of the extracellular fluid. We have a selective permeability in the cell membrane to various ions, so ions can move across that membrane. And there are different active transport mechanisms that are also available in the membrane that can aid in this membrane potential. So we're going to start first with looking at a typical cell environment. Here we have a typical cell environment. Now, I don't want you to get confused with some of the numbers. The numbers are not as important as simply the quantity that they are. So if we look at ECF, we see 142 mm. This stands for millimolar. This is going to look at the actual molecular weight and total quantity of sodium in the extracellular fluid. So a larger number simply means a greater concentration of. So here in the ECF, we have a greater concentration of sodium than we do of potassium. We also have a good amount of chloride in the outside environment. How much is not important right now. Just know that we have a higher amount of it outside of the cell than inside of the cell. Inside of the cell, we have a higher amount of potassium and a lower amount of sodium. So this is going to make up our typical cell environment, and this is going to be important to remember. I would make note or draw a separate picture of this while you're following along so you can understand the movement of these ions, but this is at rest. To understand how each ion affects the membrane potential, we're going to isolate each ion in a little bit of a thought experiment. First, we're going to talk about sodium. Now, at rest, there is a chemical gradient that is directed into the cell. And this should be fairly obvious because we remember that sodium is higher on the outside of the cell than it is on the inside of the cell. By the simple laws of diffusion, the diffusion gradient would actually allow sodium to go inside of the cell. Now, if the membrane were freely permeable to sodium, sodium would continue to enter the cell until the membrane potential reached positive 60 millivolts. This is known as the equilibrium potential for sodium. Now, I mentioned equilibrium potential, and I threw out a term where we just talked about measuring it in millivolts. Positive 60 millivolts is what I said in the previous slide. We're actually going to measure the overall voltage that is produced by the cell from the movement of ions across the membrane. Remember that sodium is a positively charged ion, so its movement is going to make the cell more positive. Here on the left, you begin with the membrane being impermeable to everything. Then, to start the experiment, you open up sodium-specific transport channels. That is, sodium will now move freely through the protein channels, but the protein inside the cell and the chloride outside of the cell can't move across the membrane. Now, the voltmeter is going to measure the difference in electrical charge between the intracellular fluid and the extracellular fluid. The extracellular fluid is assigned a value of 0, so the final reading is the electrical potential on the inside of the cell relative to the outside of the cell. Now I want to talk specifically about potassium. and we're going to remove sodium from our minds for a moment. At rest, remember that there's a lot of potassium that is inside the cell. So that means the chemical gradient is directed out of the cell. Again, using the laws of diffusion, that diffusion gradient is going to go from high to low concentration. So from inside of the cell to outside of the cell. If the membrane were freely permeable to potassium ions, in another thought experiment, potassium would continue to leave the cell until the membrane potential reached negative 94 millivolts, which is the equilibrium potential for potassium. Here we can see the diagram for potassium. Note on the left here, potassium is in high concentration inside of the cell. And as it leaves the cell, it's going to make the cell more negative. Why? Because potassium is a cation. It is positive. So as potassium leaves the cell, it means the cell is getting more negative because those negative proteins are still there. Potassium is going to keep going out of the cell until the cell reaches an equilibrium potential of negative 94 millivolts. Now, in either of those experiments with either sodium or potassium, we kept moving the ion naturally through diffusion until it reached an equilibrium potential. Notice I didn't say an equal concentration everywhere. That's very important. So why didn't the diffusion of sodium or potassium keep going until it was equal concentration everywhere? The answer to that is because the energy needed to separate the opposite charges stopped it. Remember that to separate these opposite charges requires energy. So for example, on sodium, the movement of sodium down its concentration gradient and across the membrane separated it from its partner anion, chloride. After just a few sodium ions have moved, the energy required to separate the positive and negative charges from the sodium and the chloride exceed the potential energy of the chemical gradient. That means we are now dealing with not just a chemical gradient, but an electrochemical gradient, as you can see here. An electrochemical gradient is a form of potential energy that is determined by a combination of ions, chemical and electrical gradients. The chemical gradient is something we're already familiar with. The concentration gradient for an ion across the plasma membrane. How much of something is there? The electrical gradient is created by the attraction between opposite charges, positive and negative. So for example, our positive and negative charges in the extracellular fluid are sodium and chloride. The positive and negative charges inside the cell are potassium and proteins. There's also repulsion between like charges, so positive positive or negative negative. This is why chloride doesn't typically move into the cell, simply because it's negative and so is the cell. There's proteins inside of there. So it's going to create repulsion between those charges. Now, equilibrium potential is gained from the actual electrochemical gradient. This is the membrane potential at which an ion's electrical and chemical gradients are equal and opposite. It results in no net movement of the ions across the membrane and indicates an ion's contribution to the resting membrane potential. So if we remember, the resting membrane potential, or excuse me, the equilibrium potential for potassium is negative 94 millivolts. The equilibrium potential for sodium is positive 60 millivolts. This means that as potassium moves, the cell becomes more negative, and as sodium moves, the cell becomes more positive. All right, so now we understand what sodium does and how it moves. It's going to move from the outside of the cell into the cell. Potassium moves from the inside of the cell to the outside of the cell. We've talked about these and we've talked about their contributions. Now we're going to look at resting membrane potential and use a little bit of physics inside of a living cell. Now, if you actually poke that voltmeter electrode into a living cell, you will measure a membrane potential at rest, when the cells aren't doing anything. This is called resting membrane potential. Let's imagine back to that first picture that I showed you of our resting environment. Extracellular fluid has a high concentration of sodium and chloride ions. The cytosol, or intracellular fluid, has a high concentration of potassium ions and negatively charged proteins, abbreviated A negative. Sodium and potassium ions are the main factors in influencing membrane potential because of their movement. Proteins and chloride ions cannot move freely across the cell membrane. Now, there are uneven distributions of proteins that provides an electrical force that is involved in the sodium and potassium movement. This is what we call the resting membrane potential. This is what we call a delicious breakfast sandwich on a croissant. So what is the resting membrane potential of a typical cell? Well, it's actually negative 70 millivolts. Now, what accounts for this negative 70 millivolts? Because that seems very negative. Well, the reason it exists is because of a structure called a leak channel. These are passive channels that are in the plasma membrane that are always open. The size, shape, and structure determine which ions can pass through them. Potassium ions are going to diffuse through potassium leak channels, and sodium ions diffuse through sodium leak channels. It turns out that the resting membrane potential is primarily due to the movement of potassium. Now remember that cells have leak channels for both sodium and potassium, but the membrane is about 100 times more permeable or leakier to potassium. Therefore, a greater amount of potassium is going to leave the cell than sodium entering the cell. This means that the membrane potential is closer to the equilibrium potential for potassium, but it isn't quite there. And this is because of the sodium leak channels that are still there, allowing for some positive charge to come inside. Now, if leak channels exist and there's just this natural movement, then what causes there to actually be a balance in the system overall? Well, in order to keep an overall balance in the body, we need a pump to exchange. This is the sodium-potassium exchange pump. It ejects 3 sodium ions for every 2 potassium ions recovered from the extracellular fluid. So it does a 3-to-2 ratio. And this is what helps maintain a stable resting potential so that it never goes too far in one direction or the other. Now we can put all of the things together that we've talked about so far. Here we can see a typical resting cell. We have chloride and sodium on the outside. We have protein and potassium on the inside. Our voltmeter has poked right in and it's measuring it at negative 70 millivolts, where there's still movement happening on the cell. Potassium is leaking out of the cell, sodium is leaking in, and we can see the sodium-potassium exchange pump exchanging 3 sodium for every 2 potassium that come in and using ATP to do so. Now, this tells us overall there are 2 important factors that influence membrane potentials inside of a living cell. These factors are the concentration gradients and permeability. Generally speaking, concentration gradients don't change. We don't get a lot of sodium or potassium variation. Now, cells, that means we get most of our change happening from permeability. Cells can alter how permeable they are to a substance by opening or closing ion channels, thereby determining the actual movement of ions and the membrane potential of the cell. So remember, like I said, that membrane potential is greatly affected by permeability. So how does permeability affect that membrane potential with the movement of specific ions? Well, if we increase potassium, the cell will hyperpolarize. Hyperpolarizing means it becomes more negative. The same is true with increasing the actual permeability of chloride. It will hyperpolarize and become more negative. By becoming more negative, we increase the membrane potential. So we're moving it further away from the actual ability of the cell to become positive. This is called depolarizing. If we increase the movement or permeability of sodium, the cell can depolarize. This means the membrane potential decreases and becomes less negative or more positive. In some cases, it could even go above 0. Now, how do we keep changing these membrane potentials with the movement of ions? We often do so with different channels. Now, you can open or close gated channels to change the permeability of the cell. Now, most of these channels are going to be closed at resting potential, and they open or close in response to a specific stimulus. This changes the rate of ion movement across the membrane and therefore changes the membrane potential. There are 3 types of gated channels: chemically gated or ligand gated, voltage gated channels, and mechanically gated channels. Chemically gated channels are also called ligand-gated channels. They open when they bind with a specific chemical in the body. An example could be the receptors that bind acetylcholine at the neuromuscular junction when we talked about skeletal muscle. They are most abundant on dendrites and the cell body of a neuron. They typically, another way to look at these is also neurotransmitter responding channels. Here we can actually see our gated channel that is closed with the sodium on the outside of the cell, and we're in a resting state. From there, the neurotransmitter is actually going to come in this case acetylcholine will come in and bind to the sites onto this channel. This will cause the channel to open, thus allowing sodium to come in. The next type is voltage gated. Now, voltage gated channels are the characteristic of excitable membranes. Here, the membranes are capable of generating or spreading an action potential, and they respond to changes in voltage. So that's where we measure things with the actual voltmeter. They could respond to either voltage going up or voltage going down. And in this case, a few great examples are voltage-gated calcium channels that we talked about at the synaptic bulb of a motor neuron, where the action potential came down and the change in the electrical potential activated the actual calcium to be able to come in. But there are, of course, voltage-gated potassium, calcium, and sodium channels that all respond to different levels of voltage. Here we can actually see the voltage of the cell is negative 70 millivolts and our channel is gated. But then we change the actual voltage of the cell. We make it negative 50 millivolts. This is going to cause a change in the overall voltage of the cell, which causes the voltage gated channel to open in response to this change, thus allowing this channel to open. The last type is mechanically gated channels. Now, mechanically gated are going to open in response to a physical distortion of the membrane. This is important in sensory receptors responding to touch, pressure, or vibration. Another example of this is the RYR channel that is opened by changing the shape of the DHP receptor in the actual excitation-contraction coupling of muscular contraction. So here we can see a channel that is closed, resting membrane potential. We then apply pressure, maybe some sort of touch. This distorts the membrane and causes the actual channel to open, allowing sodium to rush in. Now, there are channel types and potentials that can be generated from these different channels. They are resting potentials that are heavily dependent upon leak channels, graded potentials that are heavily dependent on chemically gated channels, and action potentials are dependent upon voltage gated channels. This is where we start to get into neuronal activity and how membrane potential can change from beyond just a resting membrane potential. Changes in membrane potential can trigger muscular contraction, gland secretion, or the transfer of information in the nervous system. We know the resting membrane potential that we've already talked about, negative 70 millivolts. This is the membrane potential of an undisturbed cell, and all neural activities begin with a change in the resting membrane potential. And this is initiated typically by a graded potential. A graded potential is a temporary, localized change in the resting membrane potential caused by a typical stimulus, and it decreases with distance from the stimulus. A graded potential, if it's strong enough, can become an action potential. Action potentials are an electrical event that involves one location on the axon membrane that is triggered by a sufficiently large graded potential. It spreads along the surface of the axon towards the axon terminals, transferring the information. Now that we've looked at things in an abstract manner, we can put it onto a neuron. The cell body itself is going to be at a resting potential. It would then get stimulated via graded potential. If that graded potential is strong enough, it will reach the axon hillock and then perpetuate itself down the axon in the form of an action potential. So now that we've laid out the neuron, we've talked about the channels, let's actually work our way to how action potentials are entirely produced. This happens first with a resting membrane potential getting a graded potential. Graded potentials are also called local potentials. They are changes in the membrane potential that cannot spread far from the site of stimulation. So a brief change, either positive or negative, that are caused by the opening of gated channels. This could be either sodium gated channels, potassium gated channels. So here we can actually see the chemically gated ion channels are going to be on the main cell body, and then we actually have the voltage gated channels on the axon. So we need to activate those chemically gated channels first to stimulate the voltage gated channels working its way down the axon. So let's go through a graded potential at rest and work our way forward. Graded potentials are produced by chemically gated sodium channels. At rest, chemically gated sodium channels are closed. When the membrane is exposed to a chemical that opens those sodium channels, sodium ions enter the cell. This brings in a rush of positive charge and reduces the membrane potential. It shifts from the resting potential of negative 70 millivolts towards a more positive value. Remember, this is called depolarization. Opening the chemically gated potassium ion channels, however, is going to cause an opposite effect. This causes an increasingly negative membrane potential as positive potassium leaves the cell. The shift in the membrane potential to more negative than resting is called hyperpolarization and increases the membrane potential. So this is an example of those different variables in a graded potential. Here we can see on the first wave of red wave, chemical stimulus opens the sodium ion channels. From there, it depolarizes, becoming more positive. Once the stimulus is removed, then the channels will close and the sodium can be removed from the cell, and it will go through a process called repolarization. Repolarization is returning back to the resting potential. Another example could be the blue wave. Here, a chemical stimulus opens potassium ion channels, thus making the cell more negative and hyperpolarizing. Once we remove that stimulus, it will return to resting potential and repolarize again. Repolarization is simply going to be the returning to resting membrane potential. As we finish this lecture, I want you to understand one key thing about graded potentials. If you can imagine how they work, it would be simply like throwing a rock in a pond of still water. When you throw that rock in the pond of water, it's going to ripple out in all directions. So even though ions are entering at one location, it will spread in all directions. The impact on the membrane potential is proportional to the size of the stimulus. A larger stimulus opens more channels, so therefore more sodium can enter and it has a greater depolarization. So larger stimulus, also think stronger stimulus. An example for this in the real world that you could visualize would be that rock in the pond of water. You throw a small rock, you get ripples that go so far. You throw a large rock, the ripples go even further. If the ripples are strong enough, they can reach all the way to the axon hillock on the neuron, thus generating an action potential, which is what we are going to talk about in our next lecture.